Before an mRNA can be translated, it needs to be processed, and that involves the removal of introns and the joining together of exons, a process that is known as splicing. One of the most important take-home messages of this section is that the same DNA sequence can serve as an exon in one cell and as an intron in another cell. So that means that we can get multiple proteins from a single gene by using different combinations of exons. This is not the same thing as saying that in eukaryotes, one mRNA typically encodes a single polypeptide. That is true. But one DNA sequence may encode multiple proteins because of differential splicing. Differential splicing refers to the fact that nuclear RNA can be spliced in different ways to get different mRNAs that will produce different proteins because they have different combinations of exons. And the different proteins that arise from this differential splicing are known as splicing isoforms. For example, from this nuclear RNA that has 5 exons, we can get different mRNAs. One of these could have exons 1, 3, and 5, another one 1, 2, 4, and 5, and so on. And this is accomplished, as I mentioned, by treating some exons as introns. By the way, number 4 here should be blue. So alternative splicing is a way to generate more proteins from fewer genes. And the majority of genes in vertebrates display alternative splicing. Splicing involves the recognition of sequences known as splice sites, which are located at the 5' and 3' ends of introns, and different splicing factors are important for that process. Cells express different splicing factors, so this means that depending on the splicing factors that a cell expresses, they may treat some exons as introns to generate a particular splicing isoform. For example, the nuclear RNA for the structural protein type 2 procollagen is processed into different splicing isoforms in precursor cartilage or chondrocytes compared to mature chondrocytes. This difference is the result of the presence of different splicing factors. Precursor chondrocytes expresses splicing factors that bind to the 5' and 3' splice sites in both introns. But mature chondrocytes only express the splicing factors that can recognize the 5' splice site of the first intron and the 3' splice site of the second intron. So exon 2 is removed along with the 2 introns. In some cases, like this one, for example, the splicing isoforms have a very similar function. So type 2 procollagen is a structural protein present in the extracellular matrix, and both of these isoforms are types of collagen. In other cases, the functions of the isoforms are completely different. For example, the isoforms of the protein BCLX have opposing roles. BCLXL, which is the larger isoform, inhibits cell death, and BCLXS, which is the smaller isoform, induces cell death. Another thing that's illustrated in this example is that splicing isoforms may actually vary just in portions of exons, so they may include portions of a particular exon. So whereas in the previous example, an entire exon was being treated as an intron in one of the splicing isoforms by skipping 2 splice sites, here both isoforms have at least a portion of the exon. And this is because the cell has factors that recognize a different 5' splice site. So there's an alternative splice site that's being recognized in this case. Recall that mutations within introns can affect gene expression, and that's because introns can have regulatory sequences. They can contain enhancers, silencers, promoters, insulators. So a mutation within an intron can cause a gene to either be expressed when it should not be or not be expressed at all. Mutations within introns can also affect gene expression by creating new splice sites. In this particular example, a mutation creates a new splice site, and this is within the gene myostatin, which acts to inhibit muscle growth. Now, because of this new splice site, part of the intron is retained in the messenger RNA. So if you compare the wild type with the mutant, there is this gray portion that represents the intron in the mutant. Within this intron, there's actually a termination codon. So the termination codon, when the ribosome reaches this point, it will dissociate from the mRNA. Because of the presence of that termination codon, a non-functional protein is synthesized. Now, in this case, the lack of functional myostatin results in greater muscle mass and greater strength. And that's because myostatin normally tells muscle cells to stop dividing and to start differentiating. So in its absence, muscles continue to divide for a longer period of time, and as you can see, we end up with a very muscular mouse. And the same is true in humans who have this particular mutation.